All right, so we're going to be starting the 5th week for physiology. And this lecture will include muscle energy metabolism, adipose tissue, and also we're going to be looking at microbiota. The learning objectives for the lecture include the source for contraction to current muscles, as well as the types of muscle fibers we find. We'll also discuss the concept of oxygen debt. We look into adipose tissue as well as microbiota. Muscle contraction is a complex process that relies on the availability of energy in the form of ATP. And the majority of this energy is used in the walk-along mechanism. And this walk-along mechanism allows for the sliding of actin and myosin filaments. That leads to muscle contraction. Now, with that said, there's also other energy-dependent processes that are essential for muscle contraction. Now, one of these processes is the pumping of calcium ions from the sarcoplasm back into the sarcoplasmic reticulum. Again, if you don't know what sarcoplasm and sarcoplasmic reticulum is, go back, please, and refresh on that. So again, this is what will help regulate the concentration of calcium ions within the muscle cell, which, again, it's necessary for proper muscle function to occur. Now, another energy-dependent process that's involved in muscle contraction is the pumping of sodium and potassium ions through the muscle fiber. So again, this process, it's necessary for the propagation of muscle fiber action potentials. And again, if you don't know what that is, a action potential is another term for, we said we can call it what? An electrical signal, right? Now, when we find this in the muscle, we say that it is referred to as the end plate potential, the EPP. So again, without these electrical signals, communication between different parts of the muscle cell wouldn't occur, or communication between even action potential as it's coming down a neuron, right? It wouldn't. If that's absent, again, the process of contraction cannot take place. So again, both muscles and nerves are working together. And when you're looking specifically in muscle cells, this sodium-potassium ions, the regulation of it, it's essential for the action potential to occur. Our muscle fibers require energy, as you said, and that preferred source of energy is adenosine triphosphate in order for them to contract. But the concentration of ATP in the muscle fibers is very small. It's about 4 millimolars, right? And this isn't enough to maintain full contraction for a very long period of time. Usually it's enough for it to last only about 1 to 2 seconds, right? Now, in order to keep going, the ATP molecules, they end up being rephosphorylated to form new ATP. And this is what allows the muscle to continue to contract. Right, so for rephosphorylation to occur, there's several sources of energy. One source is the breakdown of creatine phosphate. Okay, and this is a molecule that's stored in muscle cells that can be quickly converted back into ATP. So the combined energy of both stored ATP as well as phosphocreatine ends up giving muscle the capacity of causing maximal contraction for about 5 to 8 seconds. So this means that during intense exercise or intense physical activity, our bodies, they have to rely on other sources of energy in order to sustain that muscle contraction. So we'll be looking at those. So they include glycolysis as well as oxidative phosphorylation. So as I said in the previous slide, another source for muscle contraction is glycolysis. And glycolysis involves breaking down glycogen that we find stored in muscle cells into pyruvic acid and lactic acid, which release energy. Now, this energy is then used to rephosphorylate ADP and generate new ATP, which can directly power additional muscle contractions and also can be used to reform phosphocreatine stores. Glycolysis is especially important during intense or long periods of exercise, as it's able to provide fast and sustained source of energy. However, there's always a downside to things. So glycolysis, it also produces lactic acid as a byproduct, which lactic acid, as you learned in the previous lecture, it can contribute to muscle fatigue as well as soreness. So glycolysis is an important mechanism for a couple of reasons. First, it's able, it can take place even in the absence of oxygen, which allows for muscle contractions to be sustained for many seconds and sometimes even up to more than a minute, even when oxygen from blood isn't available. The next thing, again, the rate of ATP formation by glycolysis is about 2.5 times as fast as ATP formation in response to cellular foods reacting with oxygen. All right, so glycolysis, it has its limitations, as many end products of the process will end up accumulating in muscle cells, which will ultimately impair the ability of glycolysis to sustain maximum muscle contraction after about a minute or so. All right, the third and the final source of energy for muscle contraction is oxidative metabolism. This involves combining oxygen with the end products of glycolysis and other cellular foodstuffs to produce ATP. So these foodstuffs include carbs, fats, as well as protein. So during prolonged maximum muscle activity, most of the energy comes from fats, but for shorter periods of about 2 to 4 hours, as much of about half that energy can come from stored carbs. Okay, so in other words, oxidative metabolism is important for providing sustained energy for long periods of physical activity. Now, anaerobic metabolism is a type of energy production that does not require oxygen. It's capable of producing more ATP per minute than oxidative metabolism. However, again, it's limited in the use and can only provide about 1 to 1.5 minutes of maximal activity. And it's mainly involved in high intensity, short duration activities like you're sprinting, okay, or you're doing some very heavy weightlifting. So therefore, it's important for activities that require quick bursts of energy, but again, it can't sustain prolonged physical activity. Now, aerobic metabolism, all right, this is a type of energy production that requires oxygen. Okay, so aerobic metabolism, it needs oxygen. It's capable of producing less ATP per minute than anaerobic metabolism. However, it's able to produce ATP indefinitely, which means it can sustain prolonged, very long periods of physical activity. And is mainly involved in low intensity, long duration activities like jogging or cycling, okay, or swimming for that matter. So again, aerobic metabolism is important for activities that require endurance as well as stamina. So based on the last couple of slides, what would suit this athlete best and what would suit this athlete best? Which one of the two mechanisms that we discussed? Carbohydrate metabolism is a process by which the body breaks down carbs, specifically glucose, to produce energy. And glucose is an important energy source for the brain as well as the red blood cells, as able to generate energy through both anaerobic as well as aerobic pathways. So the metabolism of glucose involves 4 main pathways. Right? First, there's glycolysis. Then there's the intermediate reaction for pyruvate to acetyl coenzyme A. Then there's the Krebs cycle, or sometimes you can also call it the TCA cycle. Okay? And then finally, there's the electron transport chain. All right? So these pathways, they work together to produce ATP. And this is the molecule that provides energy to the body cells. So glycolysis is the first step in this process, and it involves breaking down glucose into pyruvate. Right? Pyruvate will then enter the Krebs cycle, where it ends up being further broken down to produce a little bit more ATP. Right? And then finally, we have the electron transport chain, and this uses the energy that's produced in the TCA cycle as well as glycolysis to create a large amount of ATP through oxidative phosphorylation. So again, in glycolysis and the Krebs cycle, we end up producing NAD and FADH, NADH and FADH, which ultimately then end up being used in the electron transport chain to produce lots and lots of ATP. So again, by the time you go through all these steps, remember, all these things, they'll happen one after the other, only when there is oxygen present. Okay, so you have to have oxygen. If you don't have oxygen, you're not going to be able to go through all these. Without oxygen, you go over here and then you get stuck over here. Okay, pyruvate cannot be converted into acetyl coenzyme A. Pyruvate will then accumulate, and then it'll end up getting converted into lactic acid. All right, so in glycolysis, you only get 2 ATP and 2 NADH. That'll be present. But again, you end up taking that into the citric acid cycle, then you end up getting more FADH and NADH, as well as a little bit, and some more ATP. Then finally, all that NAD and FADH that was produced in these couple of steps will then end up being yielding a large amount of ATP, up to 38 ATP molecules. All right. Again, glycolysis is the first step in breaking down glucose to form ATP. Okay, so it takes place in the cytosol of the cell and it involves a 10-step process. We're not going to go into all these steps, but very simplistically, during glycolysis, one of these glucose molecules ends up being converted into 2 molecules of pyruvate. Okay, also you'll produce 2 molecules of ATP, right? So it also produces 2 NADH, and then you also get a couple of hydrogen ions, right? So 2 water molecules are also produced as byproducts, by the way. And that is all glycolysis. You don't need oxygen for this, okay? All right, so what is this Corky cycle? Or I'm sorry, not Corky, Cori cycle. So this is a metabolic pathway in the liver that plays a key role in maintaining blood glucose levels. And it involves the conversion of lactic acid that's produced by muscles during anaerobic metabolism into glucose, which can then be used by muscles as an energy source. So the glucose is then transported back to the muscle for use in glycolysis. So again, this Cori cycle plays an important role or process during periods of intense exercise when the muscle is relying on anaerobic metabolism and is producing large amounts of lactic acid. Right. So again, remember, when there's no oxygen present, the pyruvate in glycolysis, what's happening? You're producing 2 ATP and you're producing pyruvate. The pyruvate then ends up getting turned into lactic acid if there's no oxygen. So when that happens, that lactic acid will then end up going to the liver, and then it'll end up getting converted back to glycogen, to glucose, and then it'll get sent back to muscle. Okay? And again, so this will take place usually when the oxygen debt has been met. And we'll talk more about that. So the proportion of fast and slow fibers in a muscle determines its contraction speed and endurance capacity. Muscles that are made up mostly of fast fibers, they react quickly, but they get tired quickly also. When you compare that to those that are made up mostly of slow fibers, they react slowly, but they're able to maintain their contraction for a much longer period of time. So the proportion of fast and slow fibers in muscles, they could be influenced by factors such as genetics, exercise, as well as age. All right, so let's take a closer look at these slow fibers. They're also known as type 1 or red muscle fibers, and they're smaller in size, and they're also innervated by smaller nerve fibers. They have a more extensive blood vessel system as well as capillaries to supply them with extra amounts of oxygen. They contain a large number of mitochondria that helps to support high levels of oxidative metabolisms, and also they have large amounts of myoglobin. And if you think back, we discussed in previous lectures, myoglobin is it's a protein that's found. It's an iron-containing protein that's found only in skeletal muscles, and it stores oxygen until it's needed. So the presence of myoglobin also speeds up the oxygen transport to the mitochondria. So these characteristics, they end up giving muscles, these slow muscles, a reddish appearance, hence the name red muscle. Okay. Moving on to fast fibers, or type 2, sometimes they're also called white muscle fibers. They're characterized by their large size as well as their strength of contraction. They have an extensive sarcoplasmic reticulum that allows the rapid release of calcium ions to initiate contraction, and they also contain large amounts of glycolytic enzymes for the rapid release of energy through the glycolytic process. Now, these fibers, they have a less extensive blood supply and fewer mitochondria, as oxidative metabolism is of secondary importance for them. They also don't have as much myoglobin in these fibers. We don't find too much. There's a deficit, there's a deficiency of the myoglobin, and this is what gives them this white appearance. Okay, moving on to fast oxidative glycolytic fibers, or FOG. FOG fibers are a type of muscle fiber that have the ability to produce energy through both oxidative as well as glycolytic metabolic pathways. So what this means is that these FOG fibers, they're able to contract quickly just like fast glycolytic fibers, but also they have some resistance to fatigue, like slow oxidative fibers do. FOG fibers would be recruited during activities that require both power as well as endurance, like middle distance running or swimming or even cycling. So again, these type of activities involve a combination of high intensity bursts of activities as well as sustained periods of activity at lower intensities. So FOG fibers, when you look at them, you'll observe that they have a high density of mitochondria, and this is what allows them to generate the energy through oxidative metabolism. In addition to that, they also have a high concentration of myoglobin. Okay, so again, myoglobin is going to be storing oxygen. That's going to help fuel oxidative metabolism. So when you compare them to the fast glycolytic fibers, remember, the fast glycolytic fibers, they rely nearly almost exclusively on glycolysis for energy production. So, they have fewer mitochondria and myoglobin, so which makes them better suited for short bursts of intense activity. But also, again, it makes them more prone to fatigue. So, fast glycolytic, again, it's you can kind of think of it as that it has the best of both worlds. So again, overall FOG fibers, you can consider them to be an intermediate type of muscle fiber that's showing characteristics of both slow oxidative as well as fast glycolytic fibers. They're able to contract quickly and generate a lot of force, but also they have some, they also have endurance in them, right? And again, the endurance, it comes because they're able to produce energy through oxidative metabolism. So this slide, it goes over all three types of the muscle fibers: slow oxidative, then we talk about the fast glycolytic fibers, and then as well as the intermediary, the FOG fibers. So be sure you look at these, and then you'll be able to see the differences amongst the different types. Right, let's talk about oxygen debt. So after intense exercise, the body's demand for oxygen increases to help with several processes. And the first of these processes is to convert these accumulated lactic acid back into glucose. Okay? And the second is to convert AMP and ADP back into ATP. Okay? Again, remember, this is used for energy. And the third is to convert creatine and phosphate back into phosphocreatine, which helps generate ATP during muscle contraction. So finally, the body needs to restore normal levels of oxygen that are bound with the hemoglobin as well as with myoglobin. So all of this extra oxygen consumption after exercise, we call that repaying the oxygen debt. All right, now we're going to be moving on to adipose tissue. We'll be looking at its definition, its function, the type of adipose tissue we find. We'll talk a little bit about lipogenesis, lipolysis, and hormones that are related to it. So adipose tissue, it's commonly known as body fat, and it serves many functions in the body. It provides insulation, thermal insulation, to help maintain body temperature. It also acts as a cushion, right, to protect organs. Mechanically, it'll help absorb any shock. Right? Let's think of it like that. And it also serves as a energy reserve. Okay, and whenever you need it, that adipose tissue can be broken down to liberate energy. Adipose tissue also functions as an endocrine organ. Okay? It secretes hormones and other signaling molecules that regulate metabolism as well as other physiological processes. In addition to that, adipose tissue, it has an inflammatory function and it can contribute to the body's immune system. So again, recall back to your first term anatomy and first term physiology class that adipose tissue is a type of connective tissue. It stores fat, and we can classify adipose tissue into two types based on the tissue color. There's white adipose tissue and there's brown adipose tissue. The first one, WAT, it appears white or yellowish, and this is the main site of energy storage in the body. While BAT, or the brown one, it appears brown, and this is due to the high concentration of mitochondria, and it's also involved in thermogenesis, in heat production. So WAT and BAT, they have different metabolic as well as endocrine functions, and they play distinct roles in the regulation of energy homeostasis in the body. All right, white adipocytes, okay, or WAT. Again, this is the most common type of adipose tissue that we have in our body. They're generally round or spherical in shape, and they contain a single large lipid droplet. And this pushes the nucleus and other organelles to the edges, to the periphery of the cell. So white adipose is located throughout the body, including under the skin, your subcutaneous region, and also around internal organs. Again, its main function is to store energy in the form of triglycerides. Now, BAT, on the other hand, again, this is a type of tissue that contains multiple lipid droplets and a high concentration of mitochondria, giving the cell this brownish appearance. Now, unlike white adipose tissue, which mainly stores energy, brown adipose tissue mainly participates in thermogenesis. Again, thermogenesis is the process of generating heat in the body. So these brown adipocytes, they're located in separated pockets in specific regions of the body, such as the paravertebral, the paravertebral, supraclavicular, as well as the periadrenal areas. Right, so adipose tissue plays a critical role in energy homeostasis. It serves as a primary site for the storage of extra energy in the form of triglycerides, or TAGs. So when energy demands are high, such as when you're fasting, right, or exercise, the adipocytes, they will release that stored fatty acids to other tissues that will end up using it as a fuel source. So triglyceride is actually an ester derived from glycerol, along with 3 fatty acids. Okay, so triglyceride, we have what? It's in the form of an ester, and it has 3 fatty acids. Okay, so again, this is how the name comes from, tri and glyceride. Okay, so triglycerides are the main constituents of body fats in humans as well as other vertebrates, okay, animals that have a backbone. And also in addition to that, even in vegetable fats as well. Fatty acids, they're stored primarily in adipocytes as triacylglycerol. Okay, triacylglycerol. This is the form that fatty acids are stored. Okay, triacylglycerol, it has to be hydrolyzed to release the fatty acid. Adipocytes, again, we find concentrations of it for us in our abdominal cavity. Right. So if you look at our gut, the reason that most people do not have a six-pack is because this is where adipocytes are stored. They're concentrated here. In addition to that, we also have a large amount of triglycerides, or the adipocytes under our skin, in the subcutaneous tissue. So, again, the point to take away is that the triglycerols that are stored in the adipose tissue are by far the largest sites of energy storage. Okay, adipocytes can accumulate fat in a couple of different ways. First, under normal feeding conditions, adipocytes take up dietary lipids in the form of free fatty acids from the circulation. This is made possible by the action of an enzyme called lipoprotein lipase. Lipoprotein lipase breaks down the free fatty acids from chylomicrons and very low density lipoproteins, or VLDLs. So the adipocytes can take them up. Second, the adipocyte can also take up glucose, which is converted to glycerol and then is used as the backbone for the esterification of fatty acids to form triglycerol. Okay? So the final step in triglycerol, or TAG synthesis, is mediated by an enzyme called diacylglycerol acetyltransferase, okay, or again you can just call it DGAT. This way, adipose tissue stores body fat as TAG. Okay, here you go, right over here, and this serves as the main energy reservoir within humans and other mammals as well. So when the body needs energy and metabolic fuels are low, the adipocytes, they'll start breaking down their stored tags through a process called lipolysis. So this generates glycerol and fatty acids by the enzymatic cleavage of tags by lipases such as adipocyte triglyceride lipase, okay, ATGL, hormone sensitive lipase, HSL, and monoacylglycerol lipase, okay, or MGL. So this process releases the fatty acids into the bloodstream, where they can be taken up by other tissues to be used as an energy source. And this will occur during physiological conditions such as when you're fasting or when you're exercising, and even cold exposure. Okay? So think about these last 3 points very carefully. Lipolysis is regulated by the sympathetic nervous system. So after a meal, insulin levels increase and suppress lipolysis. However, in conditions such as diabetes and obesity, where adipose tissue becomes insulin resistant, insulin is no longer able to inhibit lipolysis effectively. So insulin also has the ability to stimulate lipogenesis. And lipogenesis is the process of making or synthesizing new fat molecules by activating LPL and increasing the transcription of lipogenic enzymes. Adipocytes secrete a number of hormones, including leptin, adiponectin, and resistin. Leptin is an appetite suppressant hormone that's involved in regulating body weight as well as energy balance. Adiponectin has insulin sensitizing and anti-inflammatory effects and plays a crucial role in glucose and lipid metabolism. Resistin has been suggested to contribute to insulin resistance as well as inflammation in obesity. So these hormones are thought to play a role in the communication between adipose tissue and other organs, including the brain, your liver, skeletal muscle, and also they think that it may contribute to the development of metabolic diseases such as obesity, type 2 diabetes, as well as cardiovascular diseases. So again, this table over here, it's showing you the hormone and it's telling you which target tissues are affected by it, its metabolic actions and other activities or functions that they have. So be sure you study this and look at this. So we mentioned adipogenesis a couple of slides ago. Adipogenesis is a process in which precursor cells differentiate and become committed to storing lipid and maintaining energy homeostasis as adipocytes. Okay? So the process is regulated by numerous factors, including nutrients, cellular signaling pathways, as well as endocrine hormones like growth hormone, insulin, estrogen, androgens, glucocorticoids, and also vitamin D. So during adipogenesis, the precursor cells, they undergo a series of complex molecular and cellular events that include proliferation, growth arrest, as well as terminal differentiation. And all these are regulated by transcription factors. So these factors, they control the expression of genes involved in lipid metabolism, adipocyte differentiation, and adipokine secretion, ultimately leading to the formation of mature adipocytes. All right, so recent evidence suggests that white adipose tissue plays a crucial role in determining whether a person develops metabolically healthy obesity or metabolically unhealthy obesity. Okay, so this is metabolically healthy obesity. This is metabolically unhealthy obesity. Right? So the properties, location, and function of white adipose tissue, or WAT, okay, are closely linked to cardiometabolic risk. So for example, WAT that's distributed subcutaneously may have a more beneficial effect on metabolic health compared to WAT that's located in the visceral region, okay, or again, the abdominal region. So additionally, the ability of WAT to store and release energy as well as its inflammatory properties may also contribute to the development of either one of these two, either metabolically healthy or metabolically unhealthy obesity. So, all right, now let's move on to microbiota. Microbiota refers to the community of microorganisms that live in a particular environment. So for us humans, example of that would be our skin, or it could be our gut, okay, in our large intestines, for example. So microbiota, they include bacteria, viruses, fungi, and other microbes. Now, on the other hand, the microbiome, okay, microbiome is referring to the collection of all the genes and genomes of microorganisms that make up the microbiota. Okay, so in other words, the microbiome covers not only the microorganisms themselves, but also their genetic material and the interactions between them. So again, we can find microbiota literally throughout our entire body, everywhere. Digestive tract, skin, mouth, nose, ears, eyes, genitals, respiratory tracts. They're found all over. Right. The gut microbiota is a community of microorganisms that live in the digestive tract of humans and animals, including bacteria, viruses, as well as fungi. Among the bacteria, the two major groups are the firmicutes and the bacteroidetes, which together make up about 95% of the total microbiota. The firmicutes, they include species such as lactobacillus, as well as streptococcus, while the bacteroidetes include species such as the Bacteroides intestinalis. All right. The remaining 5% of the microbiota is made up of other primitive bacterial groups, including proteobacteria, such as E. coli, and the actinobacteria, such as bifidobacteria. So these microorganisms, they play an important role in digestion, nutrient absorption, as well as the immune system function. And they're also being studied for their potential role in various health conditions. All right, so as I said, the gut microbiota, they play an important role in the process of digestion. So these microorganisms, they're relying on partially digested food that's coming from the small intestines for their nutrition and their survival. So they actively participate in the digestion of carbohydrates, fats, as well as proteins in the foods that we eat. They're able to break down complex molecules that our body cannot digest, such as dietary fibers, into much simpler compounds that our body can absorb and utilize. In addition to that, some gut bacteria, they produce enzymes that help in the digestion of certain food components, such as lactose in milk. So again, this is highlighting the importance of maintaining a healthy gut microbiota for proper digestion, as well as for overall health. So gut microbiota, they also play a role in vitamin biosynthesis, which is the process of producing vitamins that our body needs to function properly. So these microbes in our gut, some microbes in our gut, are able to synthesize many of the water-soluble vitamins, such as the B vitamins and essential fat-soluble vitamins like vitamin K. So again, a healthy and balanced microbial community it's essential for synthesizing these vitamins. And again, these vitamins, they can help prevent nutrient deficiencies, and they will lead to supporting your overall health, a positive effect on overall health. All right, the gut-brain axis, the gut-brain axis. This is a connection between the digestive system and the central nervous system. Remember, another name for the digestive system is the enteric system, right? So this connection allows for communication and signaling between the two systems in both directions, meaning that the gut can influence the brain and the brain can influence the gut. So the axis plays a crucial role in various physiological processes such as digestion, metabolism, as well as immune function. In addition to that, it'll affect your mood, cognition, as well as behavior. So dysfunction of the gut-brain axis has been found to be linked to various disorders, including IBS, irritable bowel syndrome, anxiety, and even depression. Now, the gut microbiota can also affect the gut-brain axis by producing metabolites such as short-chain fatty acids, and these short-chain fatty acids influence the release of hormones from the gut's neuroendocrine cells. For example, peptide YY, or you can also call it PYY, it's a satiety hormone. Satiety hormone means that it makes you feel full. You don't feel hungry, right? So again, it reduces the urge to eat, and so again, it can be stimulated by these metabolites. So this shows how the gut microbiota can play a role in regulating appetite as well as body weight. The gut microbiota has an important role in immune modulation as well, right? Meaning that it influences the body's ability to deal with disease and infections. Research has suggested that certain bacteria in the gut are involved in the development of allergies as well as autoimmune disease. So breastfed babies, they have an immunological advantage as they receive secretory antibodies from the mother's breast milk that protect them against pathogens in early infancy. Breast milk also contains beneficial bacteria such as lactobacillus and bifidobacterium, which can enhance the gut as well as systemic health. Dysbiosis, this is, so first of all, if you break down the word, anytime you see dys, it means it has a negative implication to it, bad, not good, So bio means life, right? So we're talking about something bad about life, so some condition dealing with life that's not good. So again, just I'm breaking this word down for you guys to kind of help you out. So if you're taking exams and you come across a word and you're not sure what it is, if you try to break down the word and find different parts of it, you may understand the context of what the question is asking. All right. So this term is used to describe an imbalance in the number of or the diversity of the microbiota. Again, so now we're referring to the collection of the microorganisms that are living in a specific environment. So now there's various causes of dysbiosis. Okay. In humans, for example, one of this cause for dysbiosis could be a woman who's undergone a cesarean section or C-section, right, to deliver the baby, bottle feeding, environmental factors, a poor diet, psychosocial stress, direct exposure to pathogenic organisms, right, as well as antibiotic therapies. So dysbiosis, again, it can have a negative effect on your health, including the risks of infection, inflammation, as well as other diseases. All right, continuing on with dysbiosis. So dysbiosis has been linked to several health conditions, including infections, Parkinson's disease, as well as autism spectrum disorder. Infections may occur due to an overgrowth of harmful bacteria in the gut. Parkinson's and autism spectrum disorders, they've been linked to altered gut microbiota, although the exact mechanism and the causality, they're still being investigated. They don't know. All right, so what are probiotics? Right? Probiotics, prebiotics. Probiotics are living organisms that can provide various health benefits when consumed in adequate amounts. Right? So they can be found in certain foods or supplements, and they may include bacteria or even yeasts. Probiotics can also help maintain a healthy balance of gut microbiota. They can improve digestion, they can boost the immune system, and they can even improve your mental health. Now, prebiotics, on the other hand, are non-digestible substances, right? So these non-digestible substances, they stimulate the growth and activity of beneficial gut microbiota. Okay? And they think of them as acting as food for the microbiota. They promote their growth and their multiplication. Prebiotics, you can find them in foods that are high in fiber, such as fruits, vegetables, and also as well as whole grains. And they've been linked to improved digestion, immune function, and even weight management. All right, that's it for this week. Thank you so much, and we'll see you guys next time.